Hello, you're all very welcome. My name is Phil Balsap, and in this week's lecture of Bio453, we're going to give an introduction to immuno-based diagnostics and multiplex platforms. The majority of the content is going to be focused on assays that quantify a target of interest through the use of antigen-specific antibodies. We're going to be focusing on things like solid phase platforms such as ELISAs that use polystyrene plates and other types of solid matrices. We're also going to talk a little bit about fluidic-based platforms that involves quantification of a target in a liquid stream. And that'll be focusing on the Luminex platform, which we'll talk about a little bit later. We're also going to deal with the issue that we have some platforms that analyze targets, that analyze single targets, and that would be indicated as a single-plex immunobioassay. But increasingly, companies are providing platforms that involve measuring multiple targets at the same time, and that would be referred to as multiplex platforms. We're also going to focus on the use of we're also going to focus on genomic targets. So we're going to deal with genomic hybridization assays, and we're also going to deal with PCR-based analysis. Okay, so what are the learning outcomes? The learning outcomes for this particular lecture will be to have a critical understanding of the concept of biomarkers with examples, to provide an understanding of the criteria that is needed to verify a biomarker for clinical use. You should also be able to understand the process of developing antigen-specific antibodies for use in laboratory science and diagnostics. In relation to this, you should appreciate the necessity for specificity and also sensitivity. And lastly, you should be able to describe the immuno-based methodologies that are currently used in diagnostics. So as I indicated before, we're going to look at a range of immunoaffinity platforms. We're going to look at ELISAs in the context of a solid-based platform. We're going to look at fluidic-based immunoassays. And further to that, we're going to look at multiplex-based systems, and that will involve the solid-based platform of MSD and also the fluid-based platform of Luminex. We're also going to have some degree of introduction to genomic affinity-based assays, which can be used in the Luminex platform, and we'll also discuss PCR. Undoubtedly, immunobioassays are big business in the world of biomedicine. They're used very broadly, ranging from very fundamental laboratory research right through to clinical diagnostics and prognostics. In today's lecture, we're going to give a little bit more focus towards the clinical application of bioassays. We aim to discuss how these targets or biomarkers are identified, how we then go through the process of ensuring that the biomarker target that we've selected is a sensitive and specific measure for that disease. And then we'll progress on then to talk about why bioassays are generally or can be selected as a platform to be used to measure that biomarker target. And in summary, one of the main reasons that bioassays are used in a clinical setting is that they are reliable, but also, and more importantly, that they're high throughput so that they can be used in a day-to-day laboratory setting. So before we start, I suppose we should try and define the whole concept of what a biomarker is. A range of different sources will define a biomarker in slightly different ways. For instance, the top example has a very broad definition, which refers to a broad subcategory of medical science that is objective indications of a medical state observed from the outside of the patient. And the other criteria they say is that it must be able to be measured accurately and reproducibly. A definition I prefer, which is from NIH America, is a biological molecule that can be found in a range of matrices such as blood, bodily fluids, or tissues. And this biological molecule would give you an indication of whether it's part of a normal or abnormal process, or of a condition or disease. A biomarker may be used to see how well the body responds to treatment for a disease or condition. And these would sometimes be referred to as molecular markers or signature molecules. So generally speaking, these molecules that have been implicated in disease pathogenesis or etiology are generally used as biomarkers to measure disease progression. So in terms of biomarkers or targets that might be implicated in disease, where would you find these type of targets? Well, first we have to consider the very basics of biology, if you like. So we have the encoding in the DNA, and we use that and we transcribe that to messenger RNA and subsequently translate it to a protein. That protein will then be used in a biological process, whether it's as an enzyme or a structural component of a cell, and it will mediate some function. Now, we have to remember, as a very basic, is that proteins are very much dictated by their structure. So any alteration of this structure could potentially lead to dysfunctional folding and dysfunctional function. So the structural integrity of the protein is important. But another thing I want to talk about is the importance of the level of protein expression. Okay? If the protein is overexpressed or underexpressed, that might contribute to disease. We have to appreciate that protein expression in a cell, in terms of biological processes, is a very tightly regulated process. So if you alter the level of expression of this protein, it has the potential to dysregulate the biological process, and in doing so, it has the potential to cause or contribute to disease initiation and progression. So in terms of biomarkers, what we will be interested in is the structure and functionality of the protein. And secondly, we'll be interested in the level expression of the protein and how this relates to disease. So if we look at this whole process, what we need to consider is that in order to detect whether there's an alteration to a specific protein, we may be able to measure changes that have happened in the DNA. We may be able to measure changes that have happened in the messenger RNA. And lastly, we may be able to measure changes in the protein. So what sort of changes would we be looking for? So in terms of inherited diseases, we have to consider that an individual may inherit a defective or altered copy of the DNA. So you might have single nucleotide polymorphisms that are inherited, and essentially you could then measure or get an indication of the structure of the DNA and subsequently determine whether it's mutated or not. So that would be an instance where it's inherited. But we also have to consider that throughout life we have the accumulation of mutations in DNA, and they're acquired. So in diseases like cancer, the use of mutations in the DNA may give you an indication of the type of cancer you have, or mutation in a specific gene may give you an indication of how likely the cancer is going to be aggressive or not. In terms of messenger RNA, it would generally be used to get an indication of the level of expression of that gene. And subsequently then, in terms of biomarkers, you could get a measurement of whether the protein is functional, but also, of course, whether there is excessive or underproduction of that protein. And in terms of biomarkers then, in terms of the protein function, you could measure the substrate or product that's involved in the function of the protein, or you could get an indication of the level of expression of the structural protein and its function. Okay, so in summary, this dogma of DNA right through the protein and the protein function, at any point along this process, it offers the opportunity to identify alterations to the normal functional biological process. So anywhere along this process, you have the potential to identify biomarkers that may be used clinically. So just as a quick example, you may or may not be familiar with tumor suppressor genes, but one of the major tumor suppressor genes is P53, also known as guardian of the genome. And this particular tumor suppressor gene is implicated in a wide range of cancers. Now, in normally expressed and functional P53, what will happen is if you have no or mild stress, the cell might encounter very minor damage, and what P53 will do will upregulate antioxidant genes to ensure that reactive oxygen species are nullified and that DNA is repaired, hence preventing the accumulation of mutations. Another function of P53 would be that if you have accumulation of damage, what P53 will do at that point when there's excessive damage is to say that actually it's going to upregulate a series of genes that will result in the induction of cell death. And the whole rationale there is that if you have excessive damage to DNA, it's probably better to eliminate it rather than daughter cells inheriting that mutated DNA. And subsequently that will avoid the initiation of cancer. But what happens in mutant P53 is that it does neither of those functions, and essentially when the cell encounters oxidative damage, you will get the accumulation of mutated DNA, and that accumulation of mutated DNA will eventually lead to the development of cancer. So here we have a standard marker. So we know that P53 has an important function in health, and that if you manage to get mutated P53 or altered P53 expression, then you are at increased risk of cancer. Now, generally what will happen is if someone's diagnosed with cancer, a biopsy will be taken and the sequence of P53 will be taken, and also the expression of P53 will be measured. And that will give you an indication of whether that tumor is P53 positive or negative. And what the clinician can then do is deem whether if it's P53 negative, then that will give a clinician an indication of how aggressive the cancer may or may not be. So that's an example of how something as basic as P53 can be used as a biomarker of disease, but more importantly, how it can be used as an indication of prognosis or disease severity. Okay, so we've talked about the importance of the integrity of the structure of the DNA and the integrity of the structure of the protein. But I did also mention that what's also important is the level of expression of the gene and the associated protein. Now, things that are important in terms of gene expression are things that you may or may not be familiar, but one major one is methylation. Okay, at the start of every gene, you have a regulatory and promoter region, and then you have the starting point where the gene will be transcribed. Okay, and in the promoter region, the way the cell tries to prevent gene expression or limit gene expression is to methylate the promoter region. So if you go to any given gene and the promoter region is methylated, you can be very sure that that gene is turned off. Okay, and the body has this nice ability to turn on, turn off genes through methylation. I mean, you have to consider that every cell in your body has the exact same DNA within it. The thing that makes a liver cell different from a colon cell is the level expression of the different proteins. And one of the biological controls of gene expression to make the differences between these two cells is through DNA methylation. Okay, so if DNA methylation becomes dysfunctional, then you're going to have some problems associated with it. Another way that the cell can regulate gene expression is the presence of an active repressor at the promoter region. If you have an active repressor present, then it will turn off the gene expression. Whereas if you have neither methylation nor the presence of an active repressor, then you're going to get good level of gene expression. Okay, so there are the regulatory processes that happen in terms of regulation of the DNA itself. But another form of regulation in relation to gene expression can be the structure and the structure of the chromatin or the histone. So as I said, if DNA is methylated, then it will condense the chromatin and cause transcriptional repression. Moving on to the histones. If the histone is acetylated, then you get decondensed chromatin and you get gene expression. In order to turn off gene expression, you will get histone deacetylase, and that will lead to deacetylation of the histone and switches off gene expression. The interesting thing with histone methylation, so when you get methylation of the histone, it can cause gene expression to be upregulated, but it can also cause gene expression to be switched off. And the thing that dictates whether the histone methylation will cause upregulation or downregulation of gene expression is the parts of the histone that are methylated. Okay, so that will dictate whether it's switched on or switched off. So up to now we've dealt with the use of the importance of when a gene is mutated and how that can lead to dysfunction of the final protein and how that can contribute to disease. And now we've also discussed the issues surrounding gene expression and the importance of gene expression in disease pathogenesis. Again, just to highlight that every biological process is a very tightly regulated process. So if you get alteration of normal gene expression, that will cause a disbalance or a dysfunction of that normal biological process, which can, in some cases, lead to the initiation of disease. So we talked about the dogma of DNA to RNA to protein. But another thing we want to introduce at this point, and it's becoming increasingly topical in terms of biomarkers, is the use of microRNA. MicroRNA are very short sequences of nucleotides that have the ability to inhibit the translation of RNA. So microRNAs are a point that enables the cell to very quickly respond to and downregulate the expression of a given protein target. So it may be the situation where a certain protein is upregulated, and when it's upregulated, it then forms its function, and you get the formation of the use of substrate to produce products. And it may be a very, very finely balanced process. And one of the ways that we can fine-tune this process is to use the microRNA to switch it off at the exact point for a balanced use of that protein. So overall, what we've shown here is the wide range of factors that control or modify biological function. So we're going from DNA methylation and how that impacts gene expression. We've also talked about the use of microRNAs and how that can impact gene expression. And we've also talked about the influence of histones and how that can impact gene expression. And with all of this centered around gene expression, any alteration to any one of these, whether it's DNA methylation, microRNAs, or histone modifications, they can all give us an indication of normal functional physiology. For example, there's increasing interest in the prognostic use of microRNAs in a range of cancers. Because what they found is that when they compare the level of certain microRNAs in blood between a normal healthy individual and an individual, for instance, with prostate cancer, that there appears to be significant differences. So that would be one example of how microRNAs may be a target. The same with DNA methylation. If there's a gene that we know is demethylated in normal healthy tissue, but methylated in disease, then by that rationale, we could use that to try and target as an indication of disease or disease severity. So in terms of sources of biomarkers, you have your genomic or genetic markers. So for instance, you can measure mutation in a gene that's responsible for PKU. Essentially, you detect for a mutation in a gene that's responsible for metabolism of phenylalanine, which is the main mediator of this disease. So that's one way you sequence a gene. If that particular gene is mutated, then you can for sure identify that person has PKU. Another one is Down syndrome, where the individual have 3 copies of a chromosome, and in that sense you can diagnose Down syndrome. More interestingly, and more contemporary, you have the measure of epigenetic biomarkers, such as epiprocolon. And this assay, basically, it looks for methylation markers in blood that actually give you an indication of colon cancer. So that's quite neat in that sense. One of the only ones out there. And then another one, Cologuard, which actually enables you to measure 2 methylation markers in stool that gives you an indication of colon cancer. Of course, then you have metabolic biomarkers of disease. For instance, you're looking at measurement of glucose for diabetes, albumin in terms of kidney dysfunction, or chloride in terms of cystic fibrosis. And then proteomic or protein biomarkers would be anti-TTG antibody in celiac disease, or trying to quantify the viral load in HIV infection. So you have genetic, metabolic, epigenetic, and proteomic biomarkers, and you have others, of course, emerging as well. And the interesting thing is, when you take all these sources of potential biomarkers, taking all these biomarkers together can help you get a better idea of the clinical picture. So you're talking about genomics, which we covered, transcriptomics, gene expression, proteomics, cytometrics, which would be the investigation of cell characteristics, and metabolomics. And bringing all that information together, it can give us a good clinical picture, which will help us make a clinical decision in terms of disease and how these biomarkers may impact on the clinical outcome. So now that we have an indication of where these biomarkers can be found, the next question is, how do we identify them? And one thing to consider is that there are two ways that generally this process can go down. One is hypothesis driven, where you have a good idea of what you want to look at and you have a good rationale for why you're looking at it, versus the outlook that it would be completely exploratory and to a certain degree random. Okay, and we'll cover a little bit more about this. And again, the options to go about doing this could be mass spec based methods, it could be genotyping, or it could be transcriptomic approaches. It could also be, if you're doing cytometrics, it could be flow cytometry or immunohistochemistry or that type of thing. So immunocytochemistry. So in terms of biomarker discovery, as we've talked about before, so the first thing is obviously to define the clinical need. Okay, you have a disease population, and you obviously want to identify biomarkers in relation to diagnosis or prognosis. And then in terms of discovery, one approach is to take a selection of patients and match controls, and then subsequently select a platform and biological material that you want to analyze for, or to mine, if you like, for biomarkers. You then undertake statistical analysis, which will enable you to verify any biomarkers identified and also validate the biomarkers that you've selected. So in terms of genomic approaches for identifying genomic biomarkers of interest, one way to do this is through genome-wide association studies. Okay, so in this situation, you have a normal group of individuals and a disease group of individuals. You go through the process of analyzing their DNA, and at the end of that process, what you want to do is identify parts of the DNA that differ between the normal and disease population, and from that process identify disease risk loci. So loci that looks as if it would be indicative of disease. You then go through fine mapping and really home in on your target of interest, and then you select the tissue of interest and investigate the differences in gene expression. Once you've undergone that process, you've then identified if there's any difference in gene expression. You can then take that and try and verify that your target does indeed impact on disease risk through the use of cell-based studies, through the use of preclinical animal models that may help to help support or build a portfolio of evidence to support this emerging biomarker in having a role in the disease. And once you've done that, you can then take it a step further and look at other ways of analysing the biomarker of interest, whether it be through SNP genotyping, straight gene expression, RNA-seq, DNA methylation, chromatin modification, DNA copy number, or you may even want to look at proteomics. So by going through that process, you can identify gene targets, and also you can identify gene targets and go through the process of verifying its potential role in the disease, which brings you closer to that step of identifying and validating as a biomarker for that disease. So that's one way you go about it from a genomic point of view. If you wanted to do the same sort of approach, looking at proteins or proteins of interest or other molecules of interest, you could go through the process where you have discovery using gel-based methods or gel-free based methods. And that's essentially where you go through the process of separating a whole range of components. Again, this is a very much exploratory process, and you want to identify spots of interest that either aren't spots of interest in the disease population that either aren't present in the healthy population or that aren't present in the same amounts as healthy individuals. And through this whole process, of whether it's gel-based or gel-free based approaches, you then at the end of that process identify a potential biomarker of interest. And once you've identified that biomarker of interest, you can then take it forward to go through the process of verification and validation. So, as you see on the left-hand side, discovery is that exploratory approach, identify components that look unique to the disease. You then take it to verification, where you may use a very sensitive method such as MS to identify and quantify the biomarker of interest. And then when you get to the step of validation, you might want to, this is where immunobioassays come into their own. Because once you've discovered it and verified that it does indeed appear to have an impact on disease and does appear to be a potent biomarker of interest, the next step is how do we bring this to a clinical setting? And that is where immuno-based bioassays can be extremely useful, because immuno-based bioassays have the potential to be high throughput, accurate and precise. And it's at the end of this process that you may want to employ immuno-based bioassays for the clinical diagnosis or prognosis. Now, to help give you an indication of this biomining process and how cumbersome it can be, so when you go through this exploratory process, there's the potential to get over 100 candidates. But then as you go through the verification steps, this can dwindle down to around 10 candidates. And then once you get to validation, the development of an assay that can accurately quantify it, this can really go down even further. And in fact, there are some biomarkers that get to the process of validation and subsequently fall away and essentially aren't valid for use in a clinical setting. So that gives you an indication of the narrow margins that we're having to deal with here. But in terms of criteria for the biomarker, just to identify the terms that you should be familiar with in that sense. So in terms of ensuring that your biomarker is sensitive, What it means here is you want to ensure the ability of the test to correctly identify those with the disease. So a true positive rate. Where specificity is the ability of the test to correctly identify those without the disease. So that would be a true negative rate. Another term you need to be familiar with is a positive predictive value. Positive predictive value is a calculation that a patient with a positive test actually has the disease. Whereas the negative predictive value is a calculation that a patient that tests negative actually doesn't have the disease. And I feel this figure here highlights the terms very well. So firstly, if we deal with people who are positive for the condition, those who tested positive were 20. This is for the fecal occult test. Okay, so those who were positive for condition and tested positive for 20 versus those who were positive for the disease and tested negative were 10. And sensitivity is the calculation of, or the ratio, if you like, percentage of the positive compared to the negative. And likewise, the negative people with who didn't have the disease, 180 tested positive, so it's a false positive. And likewise, 1,820 people who didn't have the disease actually tested negative, which is a true negative. So in terms of positive predictive value and negative predictive value, positive predictive value gives us an indication of the amount of individuals who test positive and who actually were positive relative to those who tested, those who were negative but tested positive.
Okay, folks, so we're going to give an introduction to the production of antibodies for clinical diagnostics, and we're going to specifically focus on the ELISA. To learn outcomes, I would expect you to have an understanding of how polyclonal and monoclonal antibodies are produced. I would expect you to be able to define and explain the range of different ELISAs. That would be the direct, the indirect, the sandwich, and the competitive ELISA. I'd also like you to be able to articulate the advantages and disadvantages of different ELISAs, and also explain the concept of lower limit of quantification and upper limit of quantification. So the whole concept of this is that in clinical diagnostics, you need to have the ability to measure target molecules, and this isn't an easy task, considering you'll want to be measuring specific targets in complex matrices such as blood, serum, urine, and cells. And these matrices have a whole range of vast amounts of different molecules such as proteins, electrolytes, and other components. And the difficulty is that you want to try and detect and measure one specific molecule within that matrix, and that's the challenge that's presented to us in the clinical laboratory. And generally speaking, analytical methods such as your mass spectrometries and other analytical methods are extremely specific and very accurate, but they're not high throughput. So it's very difficult in many cases to implement these type of technologies in the clinical lab. And the other thing is a lot of these technologies require some sort of sample preparation before you use them. So we're going to take you through why using ELISA-based technologies is a useful approach. And the whole concept is that the immune system has the ability to produce antibodies that have extremely high specificity for a given target. and it's exploiting this property of the immune system, which we're going to talk about. So all of this has led to development of antibody-based techniques that enable the identification of selected targets. And of course, the next thing then is, okay, so we detect the targets, how do we quantify them? And generally speaking, in ELISAs, the quantification will be done through the use of enzyme-mediated activity, or in other technologies, you can also have fluorescent tags attached to the antibodies, which enable quantification. But getting it right back to the basics, what is an antigen? An antigen is anything that can be recognized by the immune system. Now, this can be self, our own proteins, but it can also be non-self, okay, such as pathogenic cell wall components and things like that. Now, usually they're quite large, and they're usually proteins. But the one thing to note is that these antigens can contain many binding regions that the immune system recognizes. And these binding regions are called epitopes. Okay, so for instance, in a diagram presented, this antigen has 4 different regions that the immune system recognizes, indicated as epitope 1, epitope 2, epitope 3, and epitope 3, or epitope 4. So generally what happens is when you get exposed to an antigen like this and you get a B cell response, you'll find that there will be 4 different B cells that produce their own individual specific antibody targeted against an epitope. Now, in a response against this antigen, that means you're going to have 4 different B cells. One B cell producing an antibody specifically for one epitope. Okay, and just move on. Okay, so here we have the antigen, and here we have 4 B cells that are produced as a result of being exposed to this antigen. Each B cell produces its own unique antibody, and that antibody will be directed against an epitope. So for instance, with the bottom cell, the purple cell, it produces antibody that will specifically interact with epitope 1. Okay? And in the second scenario, the blue plasma cell will produce antibody which will be targeted against epitope 4. And then the grey plasma cell will produce antibody direct against epitope 3, and the orange will produce an antibody direct against epitope 2. So that is how the immune system will respond. Okay, and here we have each one binding. So now I'm going to talk about how this can lead to the isolation of polyclonal antibodies. Okay, so we have a target. Okay, so say for instance it is C-reactive protein. Okay, a protein in our bodies that gives us an indication of whether an inflammatory response is taking place. So we isolate a version of C-reactive protein. We modify it to convert it to this thing would be called a hapten. And the basic concept of this is that basically you're modifying the antigen to make it more readily recognized by the host. In this case, it'll be a mouse. Okay, so it's ensuring that the target will be picked up by the immune system. So once we've produced this modified antigen, we inject it into the mouse model, and we wait for a period of time, and during that period of time, the mouse will produce a cocktail of antibodies directed against CRP, or the antigen. So we allow that reaction to take place, and then we collect the serum and we extract the antibodies that's produced. Okay, now the antibody that's produced will be similar to this scenario where you have different antibodies targeting the antigen, with each one of those antigens targeting their own individual epitope. So the polyclonal, the antibodies isolated are referred to polyclonal antibodies because there are a number of different antibodies produced against different epitopes of that antigen. Okay, so that's polyclonal antibodies. Now, what if we want to isolate one of these individual antibodies or specifically? So what we do is we go through the exact same process. We modify the antigen, we expose it to the animal. The animal then creates an immune response. And when it creates an immune response, it'll produce the B cells associated with it. And what the research companies then do is they isolate an individual B cell from the spleen. Okay, they isolate an individual B cell that will produce a specific antibody. They then fuse that with a myeloma cell. And the reason for that is in order to expand or increase the number of these B cell clones, we fuse it with a cell that's immortal. Okay, so it's a cancer cell. And when you fuse it with a cancer cell, you get a hybridoma cell. Okay, and that basically makes this B cell now immortal. That means we can then continuously, over a long period of time, reproduce this isolated B cell clone. And we do that through cell culture. And when we produce sufficient numbers of these B cells, we then stimulate them to produce antibody. And the antibody that's isolated is one specific antibody directed against one specific epitope. And that's why it's called a monoclonal antibody, because it's a single antigen-specific antibody that we can then use to detect our target. Okay, so the antibody collected at the end of this process is sensitive for the detection of the original target. And in the original example, I said CRP. So if someone came into the doctors and they wanted to work out if the individual had in fact had an inflammatory response or had an infection, they would take their blood and use a kit containing this antibody that detects CRP. And that way they'll be able to quantify it and make a clinical decision. Okay, so this is essentially what happens. You have 4 clones producing the antibody response, and when we're isolating the monoclonal antibody, we only isolate one of these. Then we fuse it with a hybridoma myeloma cell to produce an immortal B cell clone that we can then progress to produce a single antigen-specific antibody.
Of course, there's advantages and disadvantages to selecting either a polyclonal or a monoclonal antibody. Polyclonal antibodies are cheaper to produce compared to monoclonal antibodies. I suppose the characteristic is the antibodies and polyclonal antibodies, the antibodies are from different B cells, whereas the monoclonal, as we spoke previously, is a single antibody from a single B cell clone. So the polyclonal is multiple different antibodies, whereas the monoclonal is a single homogeneous antibody. The polyclonal antibody will bind to different multiple sites on one antigen, whereas the monoclonal antibody will bind to a single site on the antigen. And the advantages to that is that you're able to target a specific region on the antigen. Now, disadvantage with polyclonal is you'll get batch-to-batch variation, which creates problems when you get to the clinical setting. Whereas the monoclonal has very low batch-to-batch variation, making it very much useful for clinical applications in terms of precision and accuracy and reliability. Which leads us to the conclusion that polyclonal isn't great for clinical diagnostics, whereas the monoclonal is very, very useful for clinical diagnostics. I suppose the advantage of the polyclonal is it's very tolerant of slight changes to the protein structure, such as phosphorylation or maybe some form of denaturation or modification of the protein, because there are multiple sites that the polyclonal antibodies can target. Then if you have 4 sites and 1 site gets modified, at least you still have the 3 other sites that will bond normally. The problem, of course, with a monoclonal antibody is that any slight change to the epitope or region that the antibody is trying to bind to will actually interfere with the ability of the antibody to work. Okay, and this is another sort of summary table of reiterating, I suppose, what we've said in the previous slide, in terms of the advantages and disadvantages. Okay, and this is a nice diagram, I suppose, to highlight how a monoclonal preparation would like versus a polyclonal preparation. As you can see, it's a single antibody for the monoclonal preparation, whereas the polyclonal has 4 different antibodies targeting 4 different regions of the antigen. So, bringing all this together, when you produce an antibody that's specific towards a target, it can then be used in a whole range of different ways. It can be used for ELISAs, which is widely used. It can be used for a Western blot. It can also be used for immunohistochemistry, immunocytochemistry. It can be used to quantify targets in flow cytometry. It can also be used for immunoprecipitation. And of course, then you have some separate luciferase-based quantification of proteins is also another approach that can be used. But that doesn't involve immunobioassays. Okay, folks, well, what predominantly I'm going to focus on today is the use of ELISAs to quantify biomarkers of interest. And then when we get to the Luminex-based system, it's almost like an adaptation of flow cytometry in relation to quantification of biomarkers of interest. So in terms of the immunodiagnostic section now that we're going to focus on, I'm going to give an introduction to immuno-based detection methods. As I said, primarily focusing on the ELISA. There is a link to an overview of flow cytometry for those of you who are interested. It's a previous recording that I've recorded. So it's there for you if you want to view it. But then we're going to focus on the multiplex-based systems. So whilst we'll give the basics of the ELISA, we'll then go on to talk about a specific type of ELISA, electrochemiluminescent-based multiplex ELISAs that are produced by MSD, which is an American-based company. But then we'll go on to talk about bead-based multiplex flow cytometry, which is a fluidic-based immuno-based bioassay system, and we'll talk a little bit more about this. These 2 platforms, to a certain degree, are in their infancy. They have a range of biomarker assays that are useful for research purposes, but that aren't clinically validated. Of course, then they also have other biomarkers that they have went through the processes of clinically validating.
Okay, folks, so we're going to talk about the enzyme-linked immunosorbent assay, also known as ELISA. ELISA generally takes place in a 96-well plate, and these plates contain 96 little holes or wells in the plate, so that you can measure 96 different independent reactions, as seen in the picture. But before we get on to the properties of ELISA, we have to cover the topic of antibodies, because the whole process of ELISA is based around the properties of antibodies. So as we've discussed before, antibodies have a specific affinity towards a given target, And I'm going to explain how that happens. So an antibody is made of two heavy chains and two light chains, seen with the blue being the light chain and the green being the blue being the heavy chain and the green being the light chain. But it's broken up into different parts. You have the Fab fragment, which is probably the most important fragment because it contains the antigen binding site, okay? So the antigen binding site of different antibodies will be different because the antigen binding site is designed in a way that it specifically binds with its target. Whereas the FC region is a region that's generally well conserved, okay? So all antibodies present in a human will have the same FC region structure. All antibodies in a mouse will have the same FC region, and so on. Now, in terms of antibody-based detection, what ELISA does is it measures the reaction of an antibody with a target that you're interested in. And it'll be a reaction that takes place in the wells of the plate. And the reason this is useful is that if we have a molecule of interest, for instance, C-reactive protein, which is used clinically to determine if someone has an infection, is that we could take a blood sample and measure the amount of C-reactive protein present in that blood sample. And the way we would do that with ELISA is we would isolate an antibody that can specifically bind to C-reactive protein. And the whole concept of the ELISA is to try and isolate your target of interest from all the other molecules that are present in the blood sample. Okay. And the way we're able to do that is you have the antibody antigen or target reaction, and then because it's the antibody and antigen are rigidly bound to the plastic plate, we can then undertake a series of wash steps to remove the rest of the unwanted material, okay, all the other proteins in the blood. And what we then do is we quantify the amount of antibody and antigen bound through an enzymatic reaction. Okay, so what happens then is the degree at which the enzyme reacts or converts the substrate into a product will give us an indication of how much antigen or target's present. And generally what happens is the substrate and product will be different colors. So the change in color will give us an indication of how much antigen is present. And the color change is proportional to the concentration of your target. But how do we work out exactly how much material or how much target is present? We essentially create a serial dilution of pure target to create a standard curve. And when you create a standard curve, you can then compare the amount of color created from your blood sample to the standard curve and determine the concentration. Okay, so the intensity of the signal of the unknown sample is compared to the standard curve to get an actual value. And the concentration of the target in this case would be a blood sample, and in a clinical sample is determined. And once you get the concentration of your target, you then compare it to reference ranges and determine whether it is indicative of disease or of a certain clinical situation. Okay, folks, so in summary, the ELISA is a antigen-antibody reaction that takes place in the plate that enables us to quantify a target of interest. But before we get into the specific examples of ELISA, we need to be clear on certain terminologies. Okay, so firstly we have a capture antibody. Capture antibody is usually polyclonal, and the concept of the capture antibody is to isolate and bind the antigen from the complex blood or other biological sample. Okay, so the capture antibody provides an avenue for us to bind the antigen and rigidly hold it down, immobilize it essentially. Now, it must be noted that capture antibody is generally only used in a sandwich ELISA, and we'll come on to that in a little while. So whilst the capture antibody is there to isolate the target, a detection antibody, which is usually monoclonal, okay, so it's very specific antibody, is used to bind to the target for quantification purposes, okay. So in this example, this is sandwich ELISA, which we'll cover later, but here we have a capture antibody isolating the antigen, okay. But then we have the detection antibody that binds to the antigen specifically in order to subsequently quantify it. You can also have a secondary antibody, and the concept of the secondary antibody is to bind the detection antibody to facilitate the quantification. Okay, so here you can see the secondary antibody binds to the detection antibody. And the thing to note is the secondary antibody has an enzyme conjugated to it. And the whole concept here is that once we have this series of reactions, which will be different based on different ELISAs, but we'll cover that in the next few slides. But once you have this series of reactions, the way we're able to quantify it is through this reaction, where a substrate reacts with the enzyme to produce a product. And this reaction causes a color change, and the intensity of color change tells us how much antigen is present or how much target is present. Now, in terms of other terminology you may come across if you buy an ELISA kit, is it will refer to anti-species antibody. What this basically means is it tells us where these antibodies came from, i.e. was this antibody produced in a mouse, was it produced in a cow, and it'll tell us whether the antibody is anti-human. For instance, in this case, if you're measuring a target in a human, and the antibody that is directed against anti-human, for instance CRP, then it's referred to anti-human, okay? So, if I was to use an example, if this antibody was produced in a sheep directed against anti-human CRP, then it's referred to sheep anti-human CRP antibody. Okay, so in this case I've used insulin. Okay, and if the detection antibody was produced in a mouse, okay, if the detection antibody was produced in a mouse directed against anti-human CRP, then it's referred to as a mouse anti-human CRP antibody. And for instance, the difference here is that with a secondary antibody, it has to be the secondary antibody has to be targeted against the FC region of the detection antibody. Okay, so this antibody has a specific affinity for the conserved region of the antibody. So in terms of this nomenclature, or the way we would do this, is the first thing you consider is where was the antibody produced? Okay, where did we extract the antibody from? Okay, so in this case, the antibody was produced or extracted from a sheep. The next thing is what species is the antibody targeting? Okay, in most cases, it'll be human. Okay, so in the clinical setting, all antibodies will be directed against a human target in a clinical lab. And just to remember that the secondary antibody is always anti-human, and it's always targeted at the FC region. Okay, so the secondary antibody will always target the FC region of the antibody. So in terms of signal quantification, we talked about the fact that with enzymes you can get a color change in terms of quantification, but it can be measured through a whole range of things. It can also be fluorescence, chemiluminescence or electrochemiluminescence, and if it's a standard color change, it'll be absorbance. You can also get other ways to quantify immunoassays such as fluorochromes, which we're not going to cover, and also you can use radiation in a radioimmunoassay. So now we're on to the actual ELISA methods, okay? So generally speaking, there are 4 types. You have the direct ELISA, the indirect ELISA, a sandwich ELISA, and a competitive ELISA. So the direct ELISA involves the presence, or generally speaking, involves the presence or absence of an antigen, okay? Indirect ELISAs are usually used to quantify antibodies within your blood, okay? So for instance, if you have produced an antibody against HIV, an indirect ELISA would be used to measure how much of that antibody you have produced. A sandwich ELISA is used to detect antigen, generally speaking. It can be direct or indirect, and we'll cover that. And lastly, you can have a competitive ELISA. So we're going to start off with a direct ELISA, okay? So in a direct ELISA, what happens is you add your sample containing the antigen target. The antigen target subsequently binds to the plastic plate, okay? Because you've isolated your target on the plastic plate, you simply then add on your detection antibody. And in this case, the detection antibody has an enzyme attached to it, okay? And then you add your substrate and then your substrate, and you get an enzymatic reaction. So the direct ELISA is actually quite quick because the detection antibody has the enzyme attached to it. Whilst an indirect ELISA is the same concept where you have the antigen, the company puts on the antigen and adheres to the plate, and then you add your blood sample, which contains an antibody that you're interested in, for instance, an antibody against HIV. And when it binds to the antigen, you then add a secondary antibody, okay? The secondary antibody binds to the Fc portion of the antibody from your blood, and it contains a secondary enzyme. And then at that stage, you add the substrate and you get a signal color change. So I'm just going to take you through each one of individually again, okay? Another example would be with the use of a direct ELISA is where if you had a milk sample and you suspected that it might contain enterotoxin, which could be dangerous when consuming milk, you add the sample to the well, and of course there's a whole range of other components in the well. They all attach to the well, okay? So the protein components present in the milk sample attach to the well and become immobilized. The next thing that happens is you add your detection antibody, which is colored in purple, and the detection antibody binds to the target, and subsequently when you add the substrate, you then do a wash to remove it, and you add the substrate and you get a color change. So just to go over that again, you have a milk sample which you think is suspected of having contamination containing enterotoxin, which is dangerous to health. So you add the milk sample and a whole range of proteins and other components bind and adhere to the plate. Okay, they're immobilized. And what happens then is you add a detection antibody with an enzyme attached, which is directed against enterotoxin, and it subsequently binds to the enterotoxin. You then wash away the excess unbound detection antibody, and then you add your substrate. Substrate reacts to the enzyme and you get a color change. Okay, now an indirect ELISA. So an indirect ELISA involves a primary antibody or a detection antibody, but it also involves a secondary antibody, usually that has a conjugated enzyme attached. And in terms of when an indirect ELISA would be used, A good example would be HIV infection. Okay, so people with HIV infection produce antibodies directed against HIV. Okay, and the presence of the antibody that's directed against the HIV that would be present in someone who's infected can be measured to give an indication of the presence of HIV. And indirect ELISAs are generally used to measure an antibody of interest that's present in the blood. So here we have an indirect ELISA. So generally speaking, an indirect ELISA, the company will provide the antigen attached to the plate. Okay, in this case, the company will attach the HIV virus antigen because we're interested in detecting the antibody. And generally speaking, what will happen is you then add your blood sample. Okay, and if it contains antibody direct against HIV, when you add it, the antibody will attach to the HIV, and the other antibody that's not directed against HIV will be washed off. So now we have HIV antibody immobilized in the plate because it's attached to the antigen. But at this stage, we have no idea of how much antibody is present. So we then add a secondary antibody. Okay, and the secondary antibody then is targeted towards the Fc region of the target antibody. When it binds, you get the secondary antibody binding to the antibody target, and excess unbound secondary antibody is washed off. What we then do is add substrate, and substrate reacts with the enzyme and causes a color change. So this is sort of an indication of what would happen in someone who has HIV versus someone who doesn't have HIV. Okay, and the left hand side you can see that you have the antibody from the blood attached to the antigen, then you have your secondary antibody attached. Now in someone who doesn't have HIV, you won't have the presence of the anti-HIV antibody in their blood, and as a result nothing can bind and you don't get any color change. Okay, and the ELISA itself can be actually very basic in that the clinician is just looking for some degree of color change. Okay, another form of ELISA is a sandwich ELISA. And this ELISA generally will involve a capture antibody, okay, that we discussed before. So the detection antibody is present on the plate. You then add the sample, and the target of interest will bind to the capture antibody. Then you'll add a detection antibody, okay, here, which will bind to to an alternate site on the antigen, and then you can add either a secondary antibody or a substrate. So let's get a wee bit more detail on the sandwich ELISA. So generally speaking, you add the capture antibody, okay. Then you'll add a blocking agent, and the whole concept of the blocking agent is to make sure there's no binding places on the plastic plate for other proteins. But generally, the addition of the capture antibody and the blocking step will be carried out by the company. So in most cases, this is how the plate looks when it's provided to you by the company. So next step is that you have a specific target in the blood that you're interested in, and you add the blood sample, and there's a whole range of different proteins present. And the job of the capture antibody is to isolate your target of interest. Okay, and here we go, the capture antibody's done its job, and the next step is that we need to remove all the non-specific proteins that we're not interested in. So we undergo a wash and they disappear. So now we've isolated the antigen target. The next step is to add a detection antibody. Okay, so at this point the capture antibody was just to isolate the target. Now the detection antibody is going to be used for quantification purposes. So the detection antibody is added, In this case, it's already attached with a enzyme. Okay, and the next step is simply add your substrate and you get a color reaction. Another way that a sandwich ELISA can occur is that you have all the steps of capture antibody, isolate the antigen, add a detection antibody here. So capture, detection, and then you can use a secondary antibody with the enzyme attached. So the difference between the two steps here is that the detection antibody has the enzyme and it's shorter. It's a shorter assay. Whereas in this assay, you have a detection antibody without an enzyme. And then you have the secondary antibody with the enzyme that will subsequently facilitate the color reaction. Okay? So in some ways, this is a direct sandwich ELISA and this is an indirect. Okay. And of course, you add your substrate and you get your color change. Now, we talked about the blocking step. So why is it important? Because if you don't block the binding spaces on the plastic plate, as seen here, then what can happen is some of the antibodies and proteins that you add will actually stick to the plate and you'll get a false positive, okay? So in the normal situation, when an antigen is absent, these detection antibodies should be washed away because there's no antigen. But what is actually happening without the blocking step is that the detection antibody is staying even after the wash because it's attached to the plastic, and subsequently you are going to get a color reaction. So a more common example of a sandwich ELISA is your standard pregnancy test. So we're going to start off with how the pregnancy test works when you don't have, when an individual is not pregnant, okay? So this is the basic setup of the strip or the kit. So you have two antibodies present, okay? This antibody is directed against the target, okay? That is the compound that's responsible for pregnancy. Whereas this antibody is directed against the detection antibody, okay? So this is how it happens. You add your urine. Of course, there's no sample, so the flow of the urine causes the detection antibody to start moving, okay? Now, it's passing this antibody because there's no antigen present, okay? So the detection antibody has has nothing to bind to, and therefore the capture antibody will not isolate the antigen, the detection antibody and antigen from the solution. So they move on and they eventually get captured by this antibody, okay? And what you then have is no binding here because there's no antigen, and you have binding here because the antibody, detection antibody, has traveled its way through and get captured here, okay? And that's why you'll see a blue signal, one blue signal for non-pregnant when there's no pregnancy, because you're getting the enzyme reaction here, but not here. Now, what happens when you are pregnant, okay? You have the compound HCG, which is produced upon pregnancy, and you have the detection antibody. The detection antibody binds to the HCG, and the urine flow then brings these complexes along the strip. Because there is an antigen present, the capture antibody in orange is able to isolate the detection antibody and antigen complex. Okay, they bind, and then the remaining detection antibody with nothing bound to it will also bind to the bottom capture antibody. When all that happens, you then should realize that the fact that this reaction is taking place means you get the blue signal, and you get a blue signal. So that's how a pregnancy test works. So in this case, the detection antibody, in this case, the individual is pregnant. So next we're going to talk about a competitive ELISA. Okay. So the whole concept of a competitive ELISA is the target or the antigen that you're interested in has to compete with a labeled antigen provided by the company. Okay, in terms of competing with the detection antibody. So in this particular ELISA, the detection antibody is attached to the plate, and subsequently what happens is, again, you undergo a blocking step to avoid non-specific binding. Then you add the complex mixture of blood containing the antigen of interest. Okay, now when there's no sample present in the blood, there's no target present, then you add the blood sample, but you also add the labeled version of the target provided by the company. And when there's no sample present in your blood, the labeled target provided by the company takes up all the binding sites and you get a very high signal. Okay, so addition of labeled antigen and serum, which contains no target, means that all the labeled antigen will bind to the antibody, and when you add the substrate, you're going to get a high signal. So in summary, no or low amount of target present in the blood sample will result in a high signal. No antigen present in test sample means the labeled antigen provided by the company is free to bind without any competition or interference from unlabeled target present in the patient sample. Now, what happens when your blood sample does have target present? What happens is you add the blood sample, you also add the labeled target provided by the company, and they both have to compete for the detection antibody. Okay, so what happens is the binding occurs, then you do a wash to get rid of all excess material. And as you can see here, there are 2 labeled targets and there are 3 unlabeled targets. So quite clearly this blood sample did have some of the target that we're interested in. Okay, so the target present in the blood effectively competed with the labeled antigen. And when you add the substrate, instead of getting that really high intense signal, you get a much lower signal. And the fact that less labeled antigen was able to bind to the antibody, then there's less signal produced when the substrate is added. Okay, so the higher the antigen present in the blood means that it's able to compete with the labeled antigen, which means you're going to get a lower signal. So this is a sort of a summary of how it would look. If your sample has high antigen concentration, it's able to compete with the labeled antigen and you get a low signal. If you have low antigen concentration, then there's more chance that the labeled antigen will be able to bind to the antibody and cause a high signal. And this is just an example of how a competitive ELISA would look. The higher the absorbance, the lower the concentration of your target. Okay, and likewise, the lower your absorbance, the higher your concentration of your target. Okay, folks, so this slide is to summarize the advantages and disadvantages of the different ELISAs. I'm not going to go into too much detail, but the direct is useful because it's quick and only involves one antibody step. And it also avoids any cross-reactivity of the secondary antibody because there is no second antibody in the direct ELISA. Disadvantage is the immunoreactivity of the primary antibody might be adversely affected by labeling with enzymes or tags. Okay, folks, you have to appreciate that when you try to attach an enzyme to the detection antibody or second antibody, there is a risk it might affect its binding affinity. So In a direct ELISA, there's a high risk that that might impact it. And a disadvantage is there's minimal signal amplification. An advantage of the indirect ELISA is the fact that the detection antibody or primary antibody isn't labeled with an enzyme, means there's a greater chance that maximum immunoreactivity is maintained. Disadvantage is you can get cross-reactivity with the secondary antibody, which can result in a non-specific signal. Okay, which again is why blocking is critically important. And the other thing is it's longer. It requires an extra incubation step because you have that secondary antibody. So advantage of a sandwich ELISA is it provides the highest sensitivity, because on the one hand you have the capture antibody binding the antigen, and you also have a detection antibody binding to the antigen. It's a preferred method when the antigen is not an antibody. Okay, so it's the preferred method if you're not looking to detect antibody present in blood. The other thing is, a disadvantage is requires more sample processing. In terms of competitive ELISA, generally has greater precision, accuracy, and reproducibility, and disadvantage is overall lower sensitivity and specificity. So in terms of how we quantify it, okay, so we get a color change, but how do we convert a color change into a concentration? Well, basically you have to do a standard curve, okay? So for instance, you work out, if I get an absorbance of 1, what concentration will that give me? In this case, if I get a concentration of 1, I go across, I go down, and it tells me that if my blood sample has an absorbance of 1, then it has a concentration of the target of around 200. Okay, so that's how you quantify it. The color change gives off some degree of optical density, and then you're able to relate the color change with the actual concentration. And of course, it's the opposite for the competitive ELISA. Sorry, it's the same, but it's inverse. So the higher the absorbance, the lower the concentration of the product. But in terms of determining when you can accept results from an ELISA, there are a few definitions you need to appreciate, okay? The first is the lower limit of detection. So here it is indicated here, okay? Here, along here. And it's the lowest concentration at which the detection signal can be distinguished from background noise. So if you add your sample and it has an absorbance of anything here or lower, you can't rely on the value that you get, because actually it's too similar to the background. The difference now is the lower limit of quantification is the lowest standard curve point that can be accurately used for quantification, okay? So the whole concept here is that in order to accurately determine the concentration, ideally you want it to be on this linear part of the graph, okay? So if you get a value anywhere from here and below, you can't accurately or precisely determine or reliably determine the concentration of your blood sample or urine sample, whatever it may be. Okay, so that's a lower limit of quantification. The other terminology is the upper limit of quantification. Okay, so once you get to the end of the linear part of your standard curve, after that point you can't rely on getting an accurate reading. So the upper limit of quantification is the highest point in the standard curve that can still be accurately used for quantification. Okay, so if I get an absorbance up here, say for instance, and I want to determine the concentration, I go across, I can't get an accurate value. Okay, so from using these three values, you can establish in what situation you can accept the result as reliable. So Anything, if you get an absorbance anywhere here, okay, then you can't rely on it. So this whole area of the standard curve is useless. And likewise, if you get an absorbance anywhere above here, you cannot use it to quantify the patient sample because it's above the standard curve. Okay, so essentially the region that we can work with is present in this part of the standard curve. So if you get an absorbance in your sample of here to here, you can rely on your results. So in terms of some issues you might have with the ELISA, so on the left hand side, this is the ideal scenario, okay? So you have a specific interaction between the target here and the capture and detection antibody. But what can happen is you can get cross-reactivity where some antigen that you're not interested in manages to interact with the detection and the capture antibody. And what's going to happen there is you're going to get a false positive because when you read the results, you're going to think that you are measuring this antigen when in reality you're measuring this non-specific antigen. So that's cross-reactivity. Another concept in the ELISA that you come across is interference. And interference is where your nice antigen becomes slightly modified. Okay, it might be phosphorylated or it might have some other components bound to it, which means the antibody is going to find it difficult to attach and bind to the target. And in that case, you're going to get a false negative, okay? Because the antibody can no longer specifically bind to your target. Okay, another thing to consider is that the companies usually optimize this, but you need to work out what is the optimal dilution, okay? And this can have an impact considerably when you have interferon factors, when you have interference. So when you do any ELISA, if you do a series of dilutions, you should always get the same result, okay? So you do a dilution and then you just use an equation to work out what the actual value is, okay? So you adjust for the dilution. So in this case, it's got diluted and then got diluted, but when the individual did their calculation, the result remained the same, okay? 12, 12, 12. But the problem is when you have interference is that in a neat solution, it's the same sample by the way, but this is a situation where you have interference. In the neat solution, there's too much interferon factors going on. So the companies realize, well, actually we might need to do a dilution. So they do a dilution and the dilution is having a better effect, okay? So it's closer to the 12, but it's not right there yet. It's still giving a false negative of 10. But then when they did a subsequent dilution, then they established that they were able to get an accurate measurement, okay? So in this situation, the ELISA did have interference, but the company established that if we dilute it to this degree, okay, fourfold dilution, we're able to get a reliable result, okay? So 12 versus 12. So that's one way that companies can overcome interference. Now, in terms of controlling for errors or trying to ensure that your results are true or that your ELISA has run well, you can use positive controls and negative controls, okay? So positive controls are provided by the company, and they're a situation where you add them, and when you add them to the plate, you expect to get a high reading, okay? If you don't get a high reading, then you can basically determine that the ELISA doesn't seem to have worked as well. You also have a negative control where you have nothing. You add nothing, and if you get a response, then you can determine that the ELISA hasn't worked because a negative control should never give a reaction. And of course, you could add standards with known concentrations, okay? And that's another way to know if your ELISA's work. And one other aspect that you need to be aware of is the coefficient of variance, okay? So this is why you always run a blood sample twice on an ELISA plate, because you want to make sure that you don't have any spurious results, and you would expect that if you add the same sample, if you measure the same sample twice, you should get the same result. If you don't get the same result, for instance, if you get over 15 to 20% difference between the two results, then you cannot accept the result, and the suggestion is you might need to repeat that sample. Okay, so what are the applications of the ELISA? Generally speaking, they're used for clinical diagnostics. So they're used clinically, but they're also used in the laboratory for research. They're, as we've talked before, the majority of them are used to detect presence of antibody or antigen, and also used to determine serum antibody concentrations. In terms of other applications, they can be used in the food industry to detect certain toxins or components of interest. They can be used to detect food allergens present in food, and they can also be used to detect pathogens. And lastly, they can also be used for toxicological measurement of specific drugs. Okay, folks, so that's an overview of the ELISA. If you have any queries or issues, please don't hesitate to contact me. Okay, folks, this is a radioimmunoassay. And in this scenario, we have an antibody directed against a target, say for instance it's insulin. The red indicator is insulin that would be present in serum, and the blue marker is radioactively labeled insulin that's provided by the company. And the way this concept works is you put the antibody directed against the insulin onto a plate. You then add the radio labeled insulin provided by the company to the antibody, and you get binding. So all the antibodies have radioactively labeled insulin bound to them. And the way this assay works is the insulin present in the blood sample will displace the radioactively labeled insulin. And when you then wash away the unbound insulin from the sample and the radioactively labeled insulin that was displaced, you wash them away, and what you then do is you take the antibodies that have bound and place it into a gamma counter. And the degree of which the radioactively labeled insulin was lost will give you an indication of how much insulin was present in the sample. Okay, so in this scenario, if you get a high radioactive labeled signal here, then you will get a low concentration of your target. If you get a high concentration of your target, say there's high insulin in the plasma, then you will get a low radioactive signal. Okay. So that's the basic concept of a radioimmunoassay. It's a competitive assay by which you start off with a known amount of radioactive target attached to the antibody. You then add your blood sample containing the target that you want to measure, and the target present in the blood sample displaces the radioactive label that's attached to the antibody. What you then do is, if you know the amount of radioactivity that was present at the start, the decrease in radioactivity can give you an indication of how much target was present in the blood sample. And hence that's why this assay would be considered a competitive assay, because the higher the amount of radioactivity detected, then that means there was less target in the blood sample that was able to displace it. Whereas if you have a high amount of target in your blood sample, it will displace a large amount of radioactive tracer, and therefore you'll get a decrease in radiation detection.
Okay, so up to this point, we've dealt with single plex ELISAs where there is one analyte of interest, and there's only one reaction that takes place in the well of the ELISA plate. But increasingly so, there's an increased move towards multiplex ELISAs. And the platform I'm going to talk about is mesoscale discovery, which is based on electrochemiluminescence. And essentially within each well of the plate of this platform, you'll have a number of different regions that will detect different analytes, as you can see here. So within each well there are in this case 9 or 8 different small subsections of that well that are dedicated to looking at individual analytes. And the way this platform generally works is each one of these little regions in the well will be coated with a capture antibody. And then when you add the sample, your target of interest will bind to the capture antibody. And then subsequently, you'll add the detection antibody afterwards, which will have this sulfo tag attached. And the concept is that when you put an electric current across the well, then if the individual segments have detected the target of interest, they will have this detection antibody, which will have this sulfo tag. And when you put an electric current across the plate, it will cause this sulfo tag to give out light. And based on the amount of light produced in each one of these dedicated regions of the plate, will give us an indication of how much of the analyte has been detected. Now, when the manufacturer is undergoing the process of developing these assays, they have to take into consideration all the issues in terms of interference between different analytes, etc. So what you may find is when you go to buy a multiplex ELISA from mesoscale discovery, they may say, for instance, look, we can do a multiplex for what you want to look at, but we can't mix these two analytes together because they interfere with each other. So that's a big limitation of the multiplex ELISA. And I suppose part of the difficulty with these platforms is they're very much in their infancy. And over time, their sensitivity and reliability is getting increasingly better. For instance, mesoscale discovery has a set of panels that can be used for fundamental research that they believe is generally reliable, but then they have a different level for clinically validated analytes of interest, and they have a greater degree of scrutiny over them. And that's sort of the business model of this company, that they will develop multiplex ELISAs for very fundamental research initially, with a hope eventually that they'll clinically validate it. But in the interim that they haven't clinically validated, they'll sell it for fundamental laboratory use in the labs. So in the first example, I've said that they add a capture antibody, then an analyte, then the detection antibody, the capture analyte detection. But there are other examples where they can change it up again, where they actually coat it with an antigen. So in this case, they've coated the plate with a protein of interest, in this case, a carbohydrate with interest, in this case, a virus of interest. And they may want to they may use the same platform for immunohisto or immunocytochemistry.